In this video, we're going to continue to look at the different types of pores there are. So now we're going to look at aquaporins. And these are the pores that allow the transfer of water into your cell. Again, water is a polar molecule, so they will not naturally travel through your cell membrane. And we're going to look at AQP1, the best studied aquaporin. And it's a Homo Tetrameric, so it's made out of 4 different proteins that are all the same, Homo Tetrameric. And it's a glycoprotein, so it's a protein with sugars attached to it. You have 6 transmembrane alpha helices, and 2 short helices are actually completely in the bilayer. And you have this long hourglass shape, which you have hydrophobic residues. These hydrophobic residues make the water actually go through the channel faster because of the hydrophobic effect. Water does not want to interact with the hydrophobic residues, so once in the channel, they want to get away from the channel. You also have arginine and histines that will hydrogen bond the water and will make their travel a little smoother, passing it off from hydrogen bond to hydrogen bond like we've seen with the other channels. You also have a very important asparagine residue that helps to distance out or create some space between the water to prevent proton hopping. Now, proton hopping is a phenomenon that happens in water in which a hydrogen can basically be passed from different water molecules. So, for example, if we have H3O plus here, it will give its hydrogen off to water, which will immediately pass off a hydrogen to another water, to another water, to another water. Now we have a charge here. And that actually happens inside your body. And when that happens, we see positive charge traveling way faster than it's possible for a hydrogen to travel. The reason why that's important inside of an aquaporin is that if the waters were close enough to proton hop, water wouldn't actually travel through the pore all that fast. Rather, you'll have a bunch of hydrogens either leaving the cell or entering the cell. Your pH will be thrown off, and we don't want that to happen. We want to maintain we want to maintain the pH inside our cell, so we don't want hydrogens to enter or escape on their own. So that's what that asparagine is doing. Now, another transporter that's very important is the glucose transport, or GLUT1, because glucose is our main fuel inside of our body. And there's 4 basic steps for getting glucose inside your body. So the first step, simply, glucose from outside the cell binds to the transporter. Then once in the transporter goes through a conformational change to open up its pore to the cell. So basically, when glucose binds, the pore is open to the outside of the cell. Then once bound, the pore changes so it's open to the inside of the cell. Then glucose dissociates, and then your pore needs to recover. It needs to go back to its original shape. And so there's double arrows here, and that's because this pore can actually run backwards. You can take glucose from the inside of your cell and throw it out, and we do have cells that do that. And when we're doing metabolism, we want to prevent this by adding a phosphate to glucose. That's why the first step of glucose, breaking down glucose for energy, glycolysis, is to add phosphate to glucose. In this mechanism, the way it's open on one side and then opens to the other side, that makes sure that only glucose is going through and no small molecules can go through with it or by itself. Remember, the cell is very regulated. These pores are so specific that they only allow one type of molecule to pass through to them, not others. Now, we've been talking about transport proteins, or pores, and so the transport proteins we've been talking about are usually always asymmetrical, so GLUT1 is asymmetrical, and they alternate between 2 conformations, T and R. So you can think of it the same way as hemoglobin. Here, T and R means like open to the outside of the cell or open to the inside of the cell. And for these translocation systems, we have three classes. We have uniports that will only transfer one molecule at a time, so GLUT1 is a uniport. We have symports, which will take two different molecules and transport them in the same directions. And we have antiports, which has two different molecules going two different ways. And these can either be passive transport or active transport. Again, active using energy because you're going against some kind of gradient. And we'll look at these systems a little more in depth in the next video. But for that, this is the end of that video, and I'll see you in the next one.